I am Veerangoda P. Patil from Kuvempu University studying Masters in Biotechnology.
I am Ravina Viraj from Kuvempu University studying Masters in Biotechnology.
I am Bindu KB from Kuvempu University currently studying MSc Biotechnology.
Swami TK from Kuvempu University pursuing Masters in Biotechnology. We did the project in BioEdge Solutions for an academic purpose.
Our topic is related to identification and isolation of pathogenic bacteria present in street foods.
We got a pathogenic bacteria which are harmful for the human beings from the street food.
We did a project on growth of endopathic fungi and evaluating its antagonistic activity from fungal data and its DNA barcoding. We cultured the leaf sample and got the endopathic cultures. So then we done the extraction of secondary metabolites, flavonoids and phenols. We got a good experience in BioEdge Solutions.
Got a good experience in handling lab equipments in BioEdge lab. We thankful for our guide Shruti ma'am. She guided us in a best way.
In order to isolate and identification of the pathogenic bacteria, we used molecular techniques, microbiological techniques, and also bioinformatic tools. So that we collected pani from the street food vendors in Nagasandra of Bengaluru. Then the sample was subjected to serial dilution in order to reduce the bacterial population during culturing. In rough culture, we obtain 5 different bacterial colonies. Then we go for subculturing to get pure colonies. Then gram staining was used to identify bacteria. It is a technique used to determine the gram positive and gram negative bacteria in our work. We get 3 gram positive bacteria which retains purple color stain and 2 gram negative bacteria which retains pink or red color stain. Later we go for DNA isolation by CTAB method. Then to analyze the polymorphism of the bacteria, we used RAPD technique with the help of OPC18 and OPD13 primers. According to neighbor joining tree and UP geometry, we get an idea about their polymorphic nature. In order to determine the 16S rRNA gene sequence, we go for Sanger sequencing method. This is the graph of automated 16S rRNA gene sequence. This raw sequence is converted into FASTA sequence by using bioinformatic tools. Then we go for BLAST to know about its percentage similarity of bacteria. Then we take top 10 hits of each sample and build a phylogenetic tree with the help of cluster omega of each sample. Finally, we find the bacteria of each sample. In the first sample, we get Bacillus subtilis, which has the percentage similarity of 92.04%, and the second sample is Bacillus amyloliquefacius, which has percentage similarity of 95.61%. Third sample is Bacillus licheniformis, which has 100% similarity. Fourth sample is Lysine-Bacillus macroides, which has 98.08% similarity. Then fifth sample is Klebsiella pneumoniae, which has 96.73% similarity. Thank you.
We done project on growth of endophytic fungus, evaluating its antagonistic property from plant Pongamia pinnata and its DNA barcoding studies. We use molecular techniques, microbiological techniques and bioinformatic tools. We collected Pongamia pinnata plant in Pina Industrial Area, Bangalore, and we isolated the DNA from Pongamia pinnata plant by using CTAB method. Then we done the electrophoresis for the separation of DNA bands. Then we go through PCR targeting the I2S2 gene for Sanger sequencing. We cultured the leaf sample of Pongamia pinnata plant plant leaf, got different fungal cultures, observe under the microscope, and to differentiate the morphological characters. We identify the secondary metabolites from the fungal culture. Qualitative and quantitative test was done for flavonoids and phenols. Flavonoids gives a yellow color and it shows the presence of flavonoid. Phenols produce a reddish brown color. It shows the presence of phenol.